2020-2021 by conveyance vaccines by industry. EpiVacCorona is a peptide-based vaccine against COVID-19 developed by the Vector Center of Virology. It consists of 3 chemically synthesized peptides that are conjugated to a large carrier protein. This protein is a fusion product of a viral nucleocapsid protein and a bacterial MBP protein. The third phase of a clinical trial, which should show whether the vaccine is able to protect people from COVID-19 or not, was launched in November 2020 with more than 3,000 participants. It is assumed it will be completed in August 2021. According to the vaccine developers, the peptides and the viral part of the chimeric protein should immunize people who received this vaccine against SARS-CoV-2 and trigger the production of protective antibodies. However, some experts in the field have expressed concerns about the selection of peptides for use as vaccine antigens. In addition, there are also serious concerns about the vaccine immunogenicity data, which have fueled independent civic research efforts and criticism by some experts. Meanwhile, the EpiVacCorona has received vaccine emergency authorization in a form of government registration and is available for vaccination outside the clinical trials. The vaccine delivered via intramuscular route and aluminum hydroxide serves as an immunological adjuvant. The vaccine includes 3 chemically synthesized short fragments of the viral spike protein peptides, which, according to the developers of EpiVacCorona, represent the protein regions containing B-cell epitopes that should be recognized by the human immune system. These peptides are represented by following amino acid sequences. 1. CRLFRKSNLKPFERDISTEIYQAGS. 2. CKEIDRLNEVAKNLNESLIDLQE. 3. CKNLNESLIDLQELGKYEQYIK. In the vaccine, all peptides are conjugated to a carrier protein, which is an expression product of the chimeric gene. This chimeric gene was created by fusion of 2 genes originating from different organisms, namely a gene encoding a viral nucleocapsid protein and a gene encoding a bacterial maltose binding protein. The fusion chimeric gene expressed in Escherichia coli. The sequence of the chimeric protein is available from the patent. The genetic construct of the chimeric gene also includes a short genetic fragment encoding a polyhistidine tag, which is used to purify the chimeric protein from E. coli lysate. After the purification, the protein is conjugated with 3 peptides in a way that only one variant of the peptide molecule is attached to each protein molecule. As a result, 3 types of conjugated molecules are created. Chimeric protein with attached peptide number 1, the same protein with peptide number 2, and finally the same protein with peptide number 3. All 3 types of conjugated molecules are included in the vaccine. According to the developers' publications, vaccine antigens are 3 peptides of the spike protein and a chimeric protein consisting of 2 parts. In addition, the polyhistidine tag, a short peptide that is introduced into a vaccine composition to purify a chimeric protein from a bacterial lysate, is also a vaccine antigen against which antibodies can form in those who have received the vaccine. A person vaccinated with EpiVacCorona can develop antibodies not only to the peptides of the spike protein, but also to other antigens present in the vaccine. According to Anna Popova, who is a head of the Federal Service for Supervision of Consumer Rights, Protection and Human Welfare, it takes 42 days for those vaccinated with EpiVacCorona to develop immunity. The primary screening of peptides for the search for the most immunogenic ones was carried out in animals. The level of antibodies that was triggered by each tested peptide after administration to rabbits was measured. In the test, hemocyanin protein was used as a carrier protein for the studied peptides. Further, on 6 species of animals, the vaccine was shown to be harmless in terms of such parameters as general toxicity, allergic properties, and mutagenic activity. In 4 species of animals, specific activity was shown, immunogenicity and protective properties against SARS-CoV-2. The main results of preclinical studies are published in the Bulletin of the Russian Academy of Medical Sciences. The study's development timeline was reported in Russian media in January 2021. There are currently 2 clinical trials of EpiVacCorona registered in the clinicaltrials.gov database. The trial, study of the safety, reactogenicity and immunogenicity of EpiVacCorona vaccine for the prevention of COVID-19, was registered in clinical trial database with clinicaltrials.gov identifier NCTO 47800035. Another trial with the same title was registered with clinicaltrials.gov identifier NCTO 4527575. Results of the trial that included data on 86 participants were published in Russian Journal of Infection and Immunity, indicating preliminary evidence of safety and an immune response.